meaning that if two given cells are neighbors, then they compete by way of local inhibition with local inhibitory circuits in the cortex. But if we complicate this matter a little bit more, now if we make this a little bit more complicated by imagining that these two cells, class 1 and class 2, in that figure there, that they're not single cells, but rather cell assemblies. So each of these, 1 and 2, actually represent a distributed circuit spanning different cortical areas capable of reverberating and holding activity over a given time period, regardless of any input. So if at the top there, you can see some of the cells involved in a given circuit. The dark dots represent cells across 6 different cortical areas that are strong and reciprocally connected. Given a target pattern, we'd see these light up and hold together even in the absence of any input. So now let's imagine that we have a number of these circuits, 3 or 4, and we're simulating them all simultaneously. That's what you can see in that chart there. This kind of resembles what we saw in the first figure, displaying a simplified scenario in which 2 clusters were stimulated in the receptor field at the same time. So all we're doing is taking this idea and applying it to a scenario where 1 and 2 are distributed circles rather than single cells. So when we stimulate these at the same time, what happens? Well, we can see the simulation results as a function of time on the x-axis there, and the total per assembly activity on the y-axis. And with 4 different circuits being stimulated, we can rank these fairly easily and establish which was the most active, the second most active, and so on. And we can see that the activity evolves with time. And most importantly, the key thing to take away here is we can see that they all present with some activity at first, but that right around this point here, we suddenly see one circuit win out, suppressing the other 3 circuits in the process. And that's what we're referring to with inter-circuit competition. These assemblies compete, for lack of a better term, for ignition, right? And we can see a winner-take-all mechanism at play here. Looking at this figure on the right, thinking back to attractor networks, one can then think of these 4 circuits as 4 different attractors, meaning that the system has 4 basins of attraction it could gravitate towards. Now, since we activated all these circuits at the same time, we initiated the system at the very center of the 4, meaning that noise plays a large role in the picking process here. And we'll get back to that later with when we discuss bias. But the key question then is, if this system necessitates a global inhibition process, how exactly does that work? Like, we need some central network responsible for global inhibition. So let's ask ourselves whether there's any evidence the brain has a global inhibitor circuit, right? Now, as we all know, there's this fairly old notion that the brain has to have some sort of regulatory mechanism in order to avoid hyper and hypoactivity, much like the kind we sometimes see in epileptic patients, like the brains go haywire and they just freak out. The system should theoretically just be this simple thing where we've got some sensory input entering the cortex, it generates some output, but there's also a circuit in charge of cortical self-regulation attached somewhere. In other words, some part of the brain must be receiving activity that is proportionate to the total activity within the cortex and projects an appropriate inhibitory response. So we've got this inhibitory mechanism necessitated by the nature of neuronal activity itself, and we know that it functions on the basis of competition between cortically distributed cell assembly circuits at the population level, the item level, and so then we integrate this idea into a little model. We've got cell assembly 1, cell assembly 2, and the necessity for competition suggests that there must be some third element, labeled G here, regulating inhibition. So we left to ask, is there any actual evidence of such circuitry in the brain, like biologically speaking? And as you might have guessed from the fact that I'm presenting this stuff in the first place, the answer is yes. It turns out that a series of studies identified circuits just like this between the cortex and subcortical structures, specifically the thalamus and the basal ganglia. So we know that these circuits linking cortical, striatal, and thalamic structures with strong evidence, particularly suggesting that the cortex projects towards the striatum, and then the striatum towards the thalamus, and then the thalamus back towards the cortex. So it's a loop. So if we assume that these structures exist, which they do, then that means that any cortical circuits, any associative structures within the cortex, even if they aren't neighboring, implicitly compete with other distributed circuits for activity. So next, we aim to realize this within a network, which is actually kind of simple. We start by taking a model consisting of different cortical areas, like this one, and we just add, let's say, one cell, which is plugged into the activity of each area. And it then feeds back some inhibitory signal proportionate to the activity it was fed. And then we replicate this new circuit for each cortical area, and we've successfully set up this particular type of regulatory mechanism. There are some others that are theorized, but not going to get into that right now. So as a result of this, we can explain this kind of competition at the distributed, at the population level, at the item level. So that if you have cells here which are not neighboring one another, so for example, let's say the visual cortex, if you have a circuit leading to the higher level areas, imagine here the ventral stream of vision processing, imagine that these areas are the inferior temporal lobe, okay? So there's a distributed circuit that goes from the visual cortex all the way to the inferior temporal lobe. Now these circuits don't need enablers to compete, and we've successfully answered the question of how global competition happens in the brain. But that does leave one more problem, and that's how exactly the bias in a perfectly zeroed simulation, so where it's exactly centered because we stimulated everything at the exact same time, how that works, because that's the bias mechanism of attention that this is all about. And the way we basically just conceptualize this is a top-down signal that incentivizes the favoring of one of these four attractors. So that'd be pre-activation or priming for any of the candidate circuits. It's kind of a straightforward idea. It's so straightforward, in fact, that I'll go ahead and leave that mechanism for Tara to explain. So, in summary, we've examined possible biological mechanisms for competition and bias in the brain. Competition can be implemented in the form of mutual inhibition via inhibitory interneurons at a local level, which relies on neighboring cortical columns, which have local inhibitory circuits. However, competition can also be implemented at the item level by way of a global inhibition mechanism, which we get by assuming a cortical self-regulatory mechanism running a soft winner-take-all system. And that's it for my portion.